Samara Silva. I'm a PhD student at the Federal University of Santa Catarina in Brazil. And today we're going to be talking about systems biology of nanocellulose synthesis, new approach to build a core metabolic model based on computational analysis. And since I think I'm the only speaker from South America, so I came from Brazil. The state is called Santa Catarina, and the capital is Florianópolis. It is an island, so and it's called Magic Island. So you can imagine why it's called Magic Island. We have a lot of beaches and mountains and culture. And the Federal University of Santa Catarina is a public university ranked as the 10th best university in Brazil. And we have actually 55 years of history. And the chemical engineering course has 30 years of existence. And my lab laboratory is called Integrative. It's an integrate actually, because I'm working in a chemical engineering department, but most of our work is in biology science. And we work in the fields of genomic engineering, bioinformatics, metabolic engineering, and also tissue engineering. So first to think about it, I'd like to read these words from Neda Baghetti. She is an expert in computational biology from the Northwestern University. And she said, in order to push science forward and discover enduring solutions, we must be creative and think outside the box. The easiest way to do that is to do what we're doing here, transforming our approach by bringing people with different expertise together to work as a team. Just to think about it. So my interest is to understand the bacterial cellulose synthesis, and it's synthesized by this bacteria, Gluconacetobacter hanseníae ATCC 23769, and newly proposed as Conadactylbacter hanseníae. And actually, the bacteria is a gram-negative bacteria, and we have different platforms. Actually, it's different scaffolds that we can produce with this bacteria. And we can obtain, we have here spheres and also membranes and vessels. And this we call a cocoon. It's a macroscopic structure. Actually, we're very interested to eat, in the end, maybe, to do a bioartificial organ, of course. And nanocellulose is, and despite the importance of this material for tissue engineering and medicine regenerative, there are a few words actually in omic science with this bacteria. So, systems biology aims to understand the biological systems in an integrated network, and for that we use different tools and concepts from genetics, proteomics, engineering, physics, biology, mathematics, and I really think that systems biology will usher in the next big technological wave, driving innovation and economic activity, just as we saw at the dawn of the information age. And on this context, I'm working more in the metabolic engineering, and one of the most used definitions of metabolic engineering is from Stephanopoulos, which says, a science which combines systematic analysis of metabolic pathways with other molecular biology techniques to improve cell properties. And one of the most important contributions is the emphasis of metabolic fluxes. And I start, during my master's degree, I started working in the metabolic field. And actually, my big interest now, during my PhD, is to integrate with transcriptomic analysis. So the main goal is to study the interactions between the components of biological systems and how these interactions give rise to the function and behavior of that systems. So the omic science actually deal with the global analysis of biological systems, integrate actually different fields. So here we have the proteomics, genomics, metabolomics, transcriptomics. A lot of data that we actually need to use all the bioinformatics tools to analyze all this data. And The integration of different areas of knowledge allow the advances in studies related with all these omics sciences. And we have an extensive data available now, and opens the way to use existing computational approaches and to develop new ones to extract new biological insights about the fundamental physiological process. And actually, computational methods can then be used to model biological processes based on all these integrated data. And some of the motivations of this job is, first, the bacterium ginseng produces some microstructure nanocellulose with high purity, a biomaterial of great interest for applications in tissue engineering and regenerative medicine. Second, we need professionals who know how to manipulate and analyze the extensive amount of data generated by sequencing technologies. Also, systems biology and omics techniques are approaches in expansion that will open new opportunities of knowledge, leading to new discoveries in several areas. And also, there are constantly new finds in computational tools and omics techniques, promising major contributions to an understanding of overall cellular process. So this graph demonstrates the widening gap between the cumulative number of genome scale metabolic reconstructions, so as you can see in blue, and in red we have all the publications with bacterial cellulose over the past 16 years. So you can see the widening gap, and actually this emphasize actually that we really need a metabolic model of this bacteria. So actually to understand how this bacteria produces the nanocellulose. So the core metabolic model developed can provide strategies for developing these biomaterials through a better understanding of ginseng metabolic pathways. And we present an initial draft of genome-scale metabolic reconstruction and network analysis of ginseng, a core metabolism model that capture its capabilities and nanocellulose biosynthesis functions. The constraint-based reconstruction flux analysis approach was used to analyze the core model and simulate different conditions to evaluate its consistency and representativity for cellulose biosynthesis. So the goal is actually to choose an organism, here Gluconesidobacter ginseng, and apply the principles and the information sciences so you can generate a mathematical model to investigate its metabolic behavior based on computational analysis. So as you can see here. So the major objective is the determination of metabolic bottlenecks in cellulose biosynthesis. Follow the step strategy. So first, the establishment of the core model and manual operation. Second, the application of the model for metabolic flux analysis only to evaluate the flux distribution. And third, the application of the model for flux balance analysis by adding objective functions. So these were the 4 steps that we used in the present work. So we have first the creation of the draft model using automated resources. Second, we have the manual operation, so you can use all the available tools that we have. And third was the conversion of the reconstruction to a computational model. In this, we use I'm working on a toolbox. It's called GenSys, and you can use this toolbox on MATLAB, so you can do all the simulations. And the fourth step is actually the biological and all the biochemical analysis of the network. So an initial metabolic reconstruction for ginseng was performed in accordance with a previously described protocol, this one, but adapting some steps in order to construct a representative core model. Actually, this protocol is properly structured for large-scale metabolic networks and well-studied organisms, when several experimental evidence are available. However, for organisms such as ginseng, some adaptations in the protocol were necessary for the construction of a simplified representative model. So, to understand the cellulose biosynthesis, it was built this core model with all these the main metabolic pathways. So we have all these, And this is actually our core model. It was like, this is the cell, and you have all the reactions, and until the production, the fibers, so the nanocellulose synthesis. So some of the simulations, the first one was the metabolic flux analysis. So we combine a set of measured flows to calculate the flux distribution of our reactions. And this graph shows the distribution of the carbon sources. So we use 3 carbon sources, glucose, mannitol, glycerol. But why? We use this carbon source because now we're using all these 3s in our laboratory, and we can see that with these different carbon sources, the bacteria produces a different cellulose. And it's very interesting about how the production works. And here it shows that 90% of glucose, 45.6% of mannitol, and 35.7% of glycerol, it was used for cellulose biosynthesis. And there are several experimental studies that evaluate what's the best carbon source that we can use for the production. And actually the carbon source depends on the minimal growth medium chosen. And because the cellulose synthesis flux using glucose was lower when you compare with mannitol and glycerol, we're looking for the other flux distribution. And the production is lower due to the deviation of the metabolic flux for the production of gluconic acid, as you can see here. Actually, when glucose entered on the system, the flux of gluconic acid was actually twice higher than with mannitol and glycerol. So MFA could confirm that the bacteria directly oxidize glucose in the form of gluconic acid, and it accumulates in the systems, and they actually decrease the pH of the medium. And actually the most accurate simulations are the FBA, the flux balance analysis, because here we can input all the restrictions and boundary conditions and also the objective function. So this is one of the FBA simulation, actually have a lot. And this one we chose the biomass flux as our objective function. And as you can see, by maximizing biomass production, the carbon flux for cellulose synthesis was without any production, and this is a very realistic scenario since we have different strains that don't produce cellulose. So, and as we check, the specific growth rates per hour was higher with glycerol, with 1.54 per hour. And actually, to evaluate all the consistency of the model, we did a lot of simulations to check, actually maximize external metabolites. We also did with nutrient limitations. So all these simulations were carried out so we can actually know if the model was consistent. So this work is under publication. We have all the simulations that I can show all here because we have a few minutes, but we can talk later. So some of the conclusions that we have is a description of minimal medium was essential to produce a metabolic model consistent with reality. The determination of objective functions that were considered relevant was essential to analyze the metabolic network. MFA analysis provides information about the contribution of each reaction to the metabolic process and evaluate what had been evidenced experimentally in the literature. By FBA analysis and simulations with different conditions show that the core model was able to predict the phenotype cells we show in these conditions, and the core metabolic network was representative and consistent in all limits and defined boundary conditions. So the future prospects that we have is to integrate transcriptomics analysis, which is my goal right now, and I really would like to work in this area. So I would like to integrate transcriptomics and metabolomics analysis, and actually because they are a powerful tool to build the relationship between information elements, genes, transcripts, and the different function elements, the metabolites in cells. And to the end, I don't know, I think anyone here is a chemical engineer, but if you are, you know what the level field, and I would like to read this. He wrote this in the Modeling Chemical Engineer 2002, and he said, may end up by suggesting the following modeling strategy. Always start by trying the simplest model, and then only add complexity to the extent needed. This is the $10 approach, or as Einstein said, keep things as simple as possible, but not simpler. And that's it. I would like to thank you for your attention. If you would like to talk more about all the simulations and the approach, and actually more information about the scaffold with bacterial cellulose, you can send an email. Thank you.
Questions for Samara?
Very nice talk.
Thank you.
You talked a bit about how the model simulations were done in MATLAB, and you did some optimization there. Were you using a MATLAB toolbox for your optimization, or were you coding that up?
Actually, because we built the toolbox, the GCs, actually it's a good one, it's very user-friendly toolbox, but yeah, we have all the codes.
So my follow-up question there is, when you do the optimization, are you guaranteed that you're getting a unique solution every time? Or are you finding local optima and maybe...
But actually when you do MFA, it's not that accurate, but when you do all the FBA analysis, it is. Because you have all the defined boundary conditions and restrictions. And actually I just showed when you maximize biomass reaction, but also you can maximize another objective functions, but yeah. We have BA here.
You are inconsistent.
Yeah.
I guess that was the math question. I'll ask the biology question. Did you try synchronizing the cells? I was wondering what the impact of the cell cycle is on these sort of flux rates that you see.
Sorry?
Did you try synchronizing the cells so they're all at the same stage of the cell cycle? Because I suspect that probably influences some of these fluxes that you see.
When you're doing like the MFA, I think we have some problems because we don't have all the boundary conditions which we can guarantee some reality. But when you do FBA, it is, we can like all the fluxes and then you have this, it's okay.
Okay, I guess we